Good morning, everyone. My name is Zhang. I'm the Deputy Manager at Flow Cytometry Facility here in LMB. And today I'm going to give you quite broad introduction to flow cytometry and tell you who we are and what we do. So my talk is going to talk you through our facility and tell you about what is flow cytometry, how a flow cytometer works, simply dig into the sample prep and also the application in biology. So at the moment, we have 4 staff in the facility. Pierre-Andre Patila is the head of facility. Me and Agi is a flow cytometry apprentice. William just joined us as a postdoc to work on a specific project. So we located in 3N185 and 3N180. There are 2 labs. If you ever need to find us, you're welcome to pop in and talk to us. We start with what kind of instruments at our facility. We have cell analyzers and cell sorters. These 3 are cell analyzers. We have a 2 BD LSR for Tesla, 1 equipped with 4 lasers with 16 color detection, 1 with 5 lasers, 18 color detection, and Cytoflex has 6 lasers with 21 color detection. We also have this is newly acquired 2 Sony ID7000 spectral analyzer. And this spectral analyzer equipped with 6 lasers with 184 detection. We have 4 cell sorters. On the left here is the old Synergy cell sorter, still going and works well. And we have BD Fax area fusion equipped with 6 lasers up to 18 color detection. Also, we have 2 Bigfoot spectral cell sorter equipped with 7 lasers up to 60 parameters. Just to let you know, to book sorting, we do the sort for you. You book time. You book time, staff time. Please email to CellSort for your booking. So in the email, please including as many information as possible. For example, your cell type, concentration, what are you going to look at? To book analyzer, please use Carrier Connect. We provide a training to train you on the analyzer. Please contact me or Pierre-Andre for the training if you ever need it. There are other cytometers in the market. These cytometers not only analyze the flow data, also give you some image while acquiring the data on the flow plot. Although we don't have these cytometers in-house, but they are accessible from other facilities around the campus. There are also something called mass cytometry. So different from conventional spectral cytometry, mass cytometry applies a metal-tagged antibody, which avoids the issue of spectral overlap and autofluorescence. And this allows a big panel fit into a very precious sample. And imaging mass cytometry allows the researcher to look at a spatial relationship in the tissue microenvironment. And mass cytometry are mainly used for immunology oncology study. These are you can get access to these kind of cytometers in CL UK. Please contact us. We can put you in contact if ever needed. So come back to the topic. What is flow cytometry? So flow cytometry is a laser-based technology used to analyze the characteristics of cells or particles in the flow as they pass one by one through a sensing point. By measuring fluorescent intensity, flow cytometry can tell you the expression of your cell expression of a cell surface or intracellular molecules, characterize, define different cell types, analyze cell size, also allow simultaneous multi-parameter analysis of single cells. So if we compare image cytometry and flow cytometry, we can say flow cytometry has its prongs and cones. Although it cannot tell you where the antigen is, but the biggest advantage, it can look at numerous parameters at one time. We might dig into a little bit how a flow cytometer works. I'm going to talk about fluorophores and fluorescence, and also tell you about what is components of a flow cytometer. So you may all know the fluorescent molecule absorb external energy, for example, a laser, and the molecule raised to excited state and lose energy, return to the ground state. The process of losing energy giving an emission light. So the emission light always longer than excitation light, and we call it, this creates Stokes shifts. So we have many fluorophores available on the market that tagged onto the antibody that allow you to use those to detect, for example, surface protein, the surface protein or molecules like DNA or intracellular molecules. So what kind of fluorophores they are? So first one is single dyes. As the name tells, they are a single fluorescent molecules. For example, the fixed AP, APC per CP. And also there are fluorophores composed two parts. We call it tandem dyes. So the emission from the donor excites the acceptor, and this acceptor molecule emits a longer light, creates a longer Stokes shift. Those are some examples of tandem dyes. So why we use tandem dyes, not only just use single dyes? The tandem dyes has its own, how do I say, the cones, for example, is sensitive to light and also sensitive to pH, and also the log variations, and we have to be careful of. However, the tandem dyes will increase number of parameters that can be detected at the same time. In other words, you can fit much more colors into your panel if you have very precious sample. So these are the examples to show you the dye and the tandem dye on the market from the BD. And you can see all these dyes, the emission across the spectrum can all be excited by the single laser, which is 4 or 5 nanometers. So we call it your brilliant violet dyes. And similarly, we have a range of dyes cover this spectrum can be excited by the single UV lasers. We have other fluorescent molecules to help us. This also widely used in flow cytometry experiment, fluorescent proteins. So the fluorescent protein can serve as a reporter gene and also assess the transfection efficiency, even can be used to study protein-protein interactions. These fluorescent proteins originally from nature, and they later be synthesized, and also the property of fluorescence being increased. So we have a range of fluorescent protein widely used here in LMB. So what a flow cytometer composed of? Basically, it composed a laser of multiple lasers, fluidic system, optic path, and also electronics and the computer system to store the data. So flow cytometry utilize the process of hydrodynamic focusing. Basically, when the sample injected into the sheath fluid stream, this sample are then dynamically focused into a single file. This enables a single cell pass through this sensing point one at a time. So for the optics, these are the range of excitation source. In other words, lasers we have in our flow cytometers, analyzers, and sorters. So these lasers are quite sufficient for, I would say, 99% of applications we use here in the LMB. So these lasers will be used lenses to focus onto the fluidic stream. So we also use mirrors to catch the emission light from certain fluoroforms. For example, we have several types of mirrors. And for example, the 540 nanometer dichroic short pass mirror, which means anything shorter, any light shorter than this wavelength will pass through anything. All the other light will gets deflected. And we have a bandpass filter, which means it only transmits a specific limited bandwidth while rejecting all the other wavelengths. I'll put this into the context. That would be probably easier to understand. So when a cell is injected to the stream in heterodynamic focused, and the excitation source, the laser, excites multiple fluoroforms on the cell. So this emission light will be collected by a collection length. And when it's entered the first filter, we call it dichroic 510 long pass filter. And anything longer than 510 will pass through. Anything shorter will get deflected. The signal will also be captured by this bandpass filter before enter the detection. So the rest of the light, the longer light, will again encounter the second long pass filter, and the same process repeats. So this process allows us to capture the signal, multiple signals simultaneously. So we are at exciting time of next generation of flow cytometry. Sony, we have two Sony spectral analyzer, as I specified previously. So different from conventional flow cytometers, the Sony spectral analyzer use light diffraction grater. So this grater evenly distributes all the emission light across a range of detector. So basically what means the whole fingerprints of your fluoroforms being captured rather than only bandwidth of the light. So by comparing to the single color reference, all this mixed of emission light be deconvoluted. Using an algorithm, here we use weighted least square. So why we use spectral technology? So the major advantage of spectral is to separate highly overlapped fluoroform. For example, APC LX4647 will not be able to fit them into the same panel in a conventional cytometer, but for the spectral cytometer, we can fit them into the same panel. So again, this allows you increase the panel size. Another advantage is to subtract autofluorescence. So these are the examples. This sample is an unstained light whole blood samples. You can see without autofluorescent subtraction, there's looks like a positive population that appears in the detector that detects for IgE. So because we know it's unstained, it shouldn't be any positive population being pulled out. So we know that this positive population is due to the contribution from autofluorescent. So after autofluorescent subtraction, we can see with this, we can see false positive population being cleared. This allows to reveal the true IgG population once we run the fully stained sample. The electronics of flow cytometer is to convert incoming photons to electric current. This current convert to voltage pulse. This pulse being analyzed, amplified, digitalized, and stored in the computer system. So how the pulse generated? As the cell into the laser, there's a pulse. The starting, you can see there's a pulse starting appearing. And once the cell in the middle of the lasers, it peaks. Once the cell exits the laser and the peak falls. So all this width and the height of this pulse being measured and the area is being calculated, digitalized and stored in the computer. So how the data collect and analyzed? As I was saying, talking about fluorescence, fluorophores, actually, there are two very important parameters we always including in our flow cytometry analysis. So one is for scatter. So one of them are for scatter. Once the cell enter a laser, not only the fluorescence it carries gets measured, also its own physical property also gets reflected. For example, cells get into a laser beam and the light hits the cell and diffract light in all directions. We put a detector in the forward direction, hence for scatter. So it measures the diffracted light. If their cells are uniform, so bigger cell will reflect more light and smaller cell will deflect less light. So the force scatter is related to the cell size. However, they are not definite measurement of cell size because the cells may be not uniform. Depends how they go into the laser beam, the profile will change. So we have set scatter. So the set scatter is measure cell complexity inside of cells or even on the surface of the cells. The set scatter is measured at 90-degree direction. So we can see by measuring the set scatter, we already can pull out several populations. By combining these two information, which always appears in the first plot on the flow analysis, you can see we can pull out 1, 2, 3 populations plus some debris. This is an example from a whole blood dot plot. So I already showed if you plot force scatter and set scatter even without any fluorophore attached, you already can pull out the populations. Lymphocytes are smaller with uniformed nuclear, so they located quite lower for set scatter. And the monocytes next, and neutrophils, which contains quite irregular nuclears, that reflects more light. So it appears bigger forward and set scatter. So how this fluorescent signal being captured and measured actually is similar to the forward set scatter. The cells carries a fluorophore into the laser beam. So it depends how abundant of that protein and the fluorophore gives out the signal. So more abundant proteins will carry more fluorophore, hence more fluorescent. You have a bigger pulse versus less abundant protein carries less fluorophores and hence the smaller pulse. If we combine the fluorescent and scatter information, we already can pull out, we already can study several populations and also apply the gating to tease out the subpopulation we want to study. This is the simple examples and the CD45 positive white cells and also some T cells population. All the flow cytometry data stored as flow cytometry standards, FCS files, in other words. We have these files can be read by the third-party software, which means you don't have to sit at the instrument to analyze your data. LMB has a site license for FlowJo. We have a site license plus 76 seats. So if you need to analyze your flow data, please reach out to me. I will distribute 1 seat to you. So I trying to recycle all these lessons so we don't, our cost doesn't increase significantly. But if you do need it, please reach out to us. All this software, you can use them to analyze, use their statistic tools, put plot wherever you think there's an interesting population. Also, they provide you the opportunity to look at your data in a high dimensional form. So not only analyzing the cells, we also sort cells. So this is a simple diagram showing how the cell sorting performed. If you have a sample, you stay with the sample and bring us the cell suspension. We want single cell suspension. So we apply this sample to our cell sorter. Once the cell sorter will analyze your samples first, and we determine which population we want to separate. So any jobs that contain the cells interested will pulled by this deflection plates and sort into a medium. You can take this, your sorted cells or purified cells, you can either further culture them or apply them to DNA, extraction or RNA-seq and so on and so forth. So this is how the cell sorter look at. On the upper channel, you can see your stream, your sample in the fluidic stream gets intersect with multiple lasers, and where it determines which population you want to sort. And any population, the jobs containing the cells that are interested being pulled by these plates. Actually, they are high voltage plates and gets deflected into plates, tubes, multiple different kinds of medium. Just let us know before you come to the sort. Let us know what kind of medium you want to bring. So this shows what kind of medium we can sort into Eppendorf's Falcon tubes, 15 or 50 mils of tubes. So sample prep is a key for success of flow experiment. So simple rules is rubbish in, rubbish out. We cannot play magic to make your sample suddenly become, nice. Some cells come naturally in suspension, which is quite easy. You just collect your cells and spin down, wash them, make sure you have single cell suspension, bring it to us. If the cell lines stick to the plastic, then you will need some things like trypsin acutase to trypsinize your cells, lift them up. And you have to optimize your trypsin, for example, time, concentration, because if you trypsinize too long, the cells will be tiered. If it's less trypsin, if you not enough time, then the cells will start clumping. So tissues are even more difficult. It probably needs few optimizations with us. You can use collagenase, for example, bring down your tissue samples. All this filter, this is available to filter samples. Some of them are 50 micrometer, some of them are 70 micrometer, because the channel we apply your cells to are very narrow. So their range we use here, 70 micron in diameter or 100 micron in diameter. So anything bigger than that will get blocked. So other considerations, so dilute sample to a certain concentration, that we can talk this with you to optimize the concentrations to avoid the coincidence, which means abort. So if the sample too concentrated, two events too close, they will get abort and not being analyzed. And get rid of unwanted cells, because the cytometer look at one cell at a time. If you have numerous kind of cells, but you only have a small proportion wanted cells, and everything will get measured, that will delay the time. We can also, these are Phycon lymph light and this also to help you to select the population, or even just get rid of some of the populations from your sample. That will significantly decrease the length of time we needed on the cytometer. And also including a viability tie to get out dead cells. So what is viability tie? It basically stains dead cells. And if without a viability dye, this is example, you have this non-specific this population can be seen is coming from non-specific binding. So if without a viability dye, you wouldn't know whether that actually is a positive or negative population. After applying a viability dye, you can see this population cleared out. So sometimes it's very useful to make sure you do the flow cytometry properly using a viability dye. So always including controls, flow cytometry always like the controls, as many controls as possible, negative controls, positive controls, and also there's a special type of controls called single color controls. Why? Because in flow cytometry, we have spectral overlap. That means the one fluoroform being detected by the PMT of another, you can see this fluoroform is kind of yellow called PE, and this still spill into the green detectors. This little signal actually is the false signal to this channel. So single color control is quite crucial. If you so we use single color control to assess the proportion that spill this certain fluoroform spill to other detectors. We apply something called compensation that will calculate and subtract a certain proportion of the signal, so remove this spectral overlap. This example, without compensation, the CD4 T cells and the CD19 B cells they are co-expressed, which is not, biological impossible. And with compensation, and we can see we have our T cells and the B cells are sitting in its own place nicely. So it's very crucial to do compensation and check with your single color controls. Okay. So we come to the applications examples. I will show you some of the examples we've done it here in LMB. So again, the first one is use fluorescent protein in your flow cytometry experiment. So these, the upper panel shows the blue and the green fluorescent protein. They were integrated in the plasma to assess the transfection efficiency. We can see some positive blue fluorescent protein, and these are the green fluorescent protein. These cells can be sorted and make a stable cell line. And lower panel shows the 4 fluorescent protein, TD tomato, Katushka, EFP, and TFP. They are 4 fluorescent protein representing 4 different transcription factors in the genetically engineered mice. So we can also use flow cytometry to study apoptosis. So these are the DT40 cells were incubated, were induced for apoptosis and stained with annexin 5 and properdine iodide, I call it PI. So in the early stage, the phosphatidyl protein flipped to external side of the cytoplasm. So annexin 5 binds to it, and annexin 5 also conjugated with a fluoroform. We can use this fluoroform to read out annexin 5. So this population is annexin 5 positive only, and they are the apoptotic cells. In the later stage, the membrane gets compromised, and the PI be able to enter the cell membrane, stain the nucleus. So these are the dead cells, shows double positive with annexin 5 and PI. So we can also use flow cytometry to study proliferation of cells. We load the cells with the cell tracing dye, for example, CFSE. In the mothering cells, every time they divide, the fluorescence halves, so we can trace the division cycles. And this is an example from a former postdoc. So the T cell, this example shows T helper cell development under the stimulation of cytokines for 4 days. Cell trace violet loaded at day 0. and GFP reporting L13 expression. So as you can see, as T cell device L13 expression increases, this indicating their development into Th2 cells. We can also use flow cytometry to study cell cycles as we are molecular biology labs using DNA binding dyes. So this example is from Chris Kuo from the Archaea. So you can see we use the Hurst to stain the Archaea DNA. And when Hurst interclicked into the DNA, it gives out a blue fluorescent. So if we look at in a linear scale, we could see G1S and a G2. So it is very crucial to get out the clumps and the doublets in this kind of experiment, because we only looking at the double mounted DNA, two cells clumped together when they pass the laser beam will classify as G2, which is, is misleading information. So the first plot will need to get on single cells, and then we apply to show the cell cycle profile. So this experiment, Chris also coupled the Hurst staining with a surface division protein, and we can see the surface division protein, how they expressed during the cell cycle. This is nicely displayed here. Titration is so crucial in flow cytometry. So sometimes less is more. So as here, you can see this is a DNA binding dye titration in Archaea. So at highest concentration, you don't see anything, but actually at a much lower concentration, it shows the best DNA profile. This is because the unbound DNA binding dyes could increase the background. This will decrease the signal to noise issue. So always titrate your dyes, or sometimes the antibody as well. So this example gives a bit more details in cell cycle analysis. So BRDU is a thymidine analog, and it cultured with the cells, and it can incorporate into DNA as cells enters S phase. And we use an antibody against BRDU, and this antibody is fluorescent tagged. So we will have the readout. So this experiment carried out by Guillaume in Julian Cell's lab. There's a beautiful graphs. So the papers are here if you are interested. And in this mouse embryonic stem cells with and without zinc finger protein regulator were given BRDU in culture and stained with Hurst and anti-BRDU antibody. You can see in the wild type cells shows a normal cell cycle profile. In contrast, in knockout cells shows a normal amount of DNA accumulated in the genome. This is due to the S phase arrest. So I think this paper will tell you about how this protein will affect genome instability in this cell line. We can also to look at chromosome, profile the chromosome, also even sort certain chromosome. So this chromosome was isolated from RPE cells. Chromosome usually stained by chromomycin A3 and DAPI. Chromomycin A3 inclined to bind GC content, and the DAPI inclined to bind AT content. So depending on the ratio, the GC and AT in all these chromosomes, we can pull out each chromosome. And if we find the chromosome we're interested, we can put a gate around a certain chromosome and pull that out from the cloud for sub-analysis. Also, there's interesting, unusual applications. So Anna Webb, former PhD students, use flow cytometry to assess DNA integrity in sperm cells. So she uses acridine orange. So acridine orange binds to double-stranded DNA and it fluoresces green. When it binds to single-stranded DNA, it fluoresces red. So she compares the wild type and the experimental state. We can see on the wild type, the sperm cell genome stay intact, and also in contrast, in experimental states, we can show there's a lot of single-stranded DNA in the sperms, which indicating the DNA damage. So the flow cytometry largely used in immunophenotyping. So we do a lot of immunophenotyping here in LMB as well. Again, I cannot address enough about titrating your antibody. This not only saves you money, because sometimes the manufacturer recommended concentration are probably way higher than you actually need. Also, to give you best signal to noise ratio, and if you're looking at something really dim, that's really crucial to pull out. You actually can see the dim population from the from all the other markers. This is all, again, very simple immunophenotyping flow cytometry plot from fresh whole blood. Again, we use a scatter fluorescent signal to pull out the population. And also, we can apply all the gating to tease out each population we want to see. Here we can see the T cells, B cells, natural killer cells, monocytes, and also activated stage of all these cells. We also use flow cytometry to characterize hematopoietic stem cells. So these shows 3 ways to characterize hematopoietic stem cells by using canonical markers, SLAM markers, and interestingly, use site population. Stem cells, the hematopoietic stem cells are proved to pumping out preloaded Hurst. So showing this falling signal to the side, we call it site population. This site population further characterized to be the hematopoietic stem cells by actually their surface marker. So we can sort these characterized hematopoietic cells to facilitate in vivo study. So in this paper, it showed you the hematopoietic stem cells sorted from wild-type mice and also DNA repair pathway impaired mice. And we sort those cells and they transformed back to the mice. And after 16 weeks, the researchers studied the hematopoietic stem cells reconstitution. So these are the papers if you're interested to read. We're not only looking at the surface proteins, we also looking intracellular cytokine, intracellular molecules, for example, cytokines. So this will involve permeabilize the cell membrane, fix and permeabilize to allow the antibody access to the cytokine inside the cells. So this is the example. This study combined with a cell trace violet to show the division cycle. And this experiment shows L2 and interferon gamma were produced at different stages of the T cell division. So L2 appears from the beginning and interferon gamma actually expressed the later stage of T cell division. We also can look at transcription factors in the cells. So to look at transcription factors, not only the antibody needs to get access to the cytoplasm, also they need to get access into the nuclear. So this will involve a hash detergent to permeabilize the nuclear membrane. So this study shows the many transcription factors being detected, for example, CATA3 or gamma T, and UMIS and TBET. This is coming from Illinois. So when we talking about flow cytometry, sometimes the people think, okay, we have huge, like large panel size. Nowadays, by using spectral cytometer, we can easily look up to over 30 colors at one time. So imagine if you have 2D plots, do one by one, and you have various tissues, this make analysis tedious. So actually, now we can use high dimensional analysis to interpret your data. I'm not going to get into the details of this paper. It's also produced by Noi, if you're interested to read. Basically, you can see we use dimensional reduction algorithm to plot all the information on one plot. So within each island, they represent the cells that having the same phenotype. So from each island, you can gauge on each island and going back to say what actually these common features are. So we can see you can look at different tissues and also look at the different markers. And again, the flow draw will be able to help you to do the high dimensional analysis. So we can also do calcium influx. So we're using a dye called Indo-1. It's a ratiometric calcium dyes. When bound to calcium, emits at blue, and also when unbound, emits at green. So by plotting the ratio of blue versus green against the time, we can see these naive T cells, displayed the influx of calcium in the cytoplasm after activated by the antibody linker. And this is a positive control. We also can measure the FRET using flow cytometry to detect membrane fusion. This is the Simona, a PhD student in Chang's lab, conducted this kind of experiment. It's very interesting. So DIO and DIL is a FRET pair. So each cell type load either with DIL and DIO. When the cells interact, the membrane fuse, we can see the DIO signal, which shows green here. Some of the cells are starting going to the red channel. So this is actually means the FRET occurs. That means the membrane fusion happened. We also make measuring bacteria possible in flow cytometry. The flow cytometry has its own limitations. For example, they are not that great to look at very small particles. But however, by playing with all the settings and parameters, we make it possible. However, we have to be careful about the noise which is coming from the buffer, coming from the electronics. We have to be carefully thresholding in the right place to ensure we actually can visualize the true bacteria. Here is the E. coli. The Rongzhen from Chang's lab and showed the plasmid containing antibiotic resistant gene coupled with GLP are transformed into the E. coli cells successfully. So the last but not least, flow cytometry can use for affinity selections. So this paper was conducted by the former postdoc here in LMB. So basically, he expressed a linear library in DT40 cells. So there will be a range of the protein displayed on DT40 surface, membrane surface, and these cells were allowed to interact with a target protein, which is IFN gamma, interferon gamma. So flow cytometry used to identify and sort the binders. So each time we select the binders, and these binders will go back to culture, incubate with interferon gamma again. As the DT40 mutate towards binding to interferon gamma, after a round of a selection and sorting, and eventually the the bander, the one of the bander displays high affinity to interferon gamma. This one was further evaluated by affinity assays by other methods. So that's all the applications. So there's some reference book for flow cytometry if you're interested, or pop in to talk to us. There's a lot of online resources to study flow cytometry. You can even, Google them in YouTube. There are plenty of resources. And finally, please come and talk to us. We provide training on instruments. We do cell sorting. We can help for flow experiment design, and we give you advice what kind of fluorophores you're going to use for experiment. If you're not very familiar with flow cytometry, do come to talk to us. There will always a lot of trouble later on in your experiment. So any possibility in use of flow, and you're welcome to see us. So these are the societies, flow cytometry societies. You can look up online if you're interested. We host a meeting every so often if you are like to attend, or even give a talk in our local Cambridge Cytometry Club. You are very welcome. We're always looking for speakers. That's all. Thank you very much.